Hello, everyone. Welcome back to our YouTube channel. Today, I'm going to talk about Chapter 5, Enzymes. This is for IGCSE CIE Biology 0610 students. I am Dr. Mary, and let's get started. What are enzymes? Enzymes are proteins that function as biological catalysts and are involved in all metabolic reactions. So here are two phrases that you have to understand. First, biological catalyst. What is a catalyst? A catalyst is a substance that increases the rate of a chemical reaction and not changed by the reaction, which means this is a substance, it can be used over and over again. So simply, enzymes are the things which increases the rate of the chemical reaction inside our bodies. The second phrase that you have to understand is metabolic reaction. Metabolic reactions means the reactions inside the cells or inside the living organisms. So there could be chemical reactions inside living organisms or in the actual world, in our environment. If those chemical reactions happen inside the cell, they are called metabolic reactions. How do enzymes work? An enzyme-controlled reaction involves a substrate, an enzyme, and a product. The enzyme is the thing which increases the rate of the conversion of substrate into the product. Enzymes work according to this lock and key model due to enzyme specificity. So let's look at this picture. This is the enzyme. The pink thing here is enzyme. And this surface that I just drawn is the active site, which means it is the site where most of the work is done. This shape of the active site is complementary to the shape of the substrate that it has to work on. So here you can see that these two surfaces are complementary perfectly so that they can fit just like lock and key. This is called specificity. Active site of the enzymes are specific to one particular substrate, which are the molecules that get broken down or joined together in this reaction. That particular substrate is complementary to the shape of the active site of the enzyme. If it is so, the reaction occurs faster than before, and the product is formed from that substrate. After the reaction, the product or products are released from that active site of the enzyme. Then the enzyme is free. That enzyme can be used in another reaction. So how do they meet? The enzymes and substrates are molecules which randomly move around in their place due to the kinetic energy that they have. So here you have to know that the higher the temperature, the more kinetic energy there is and the faster they move. So if we increase the temperature, the enzymes and substrates will move randomly faster and will meet each other faster so that they collide. And the substrate fit into the active site of the enzyme. Then they become an enzyme substrate complex. Then the reaction occurs. After that, product or products are formed from that substrate and they are released from the active site. The enzyme is remain unchanged and will go on to catalyze further reactions. So let's look at these two pictures. Here, the first row is about joining together two substrates. We call it building up or synthesis. It is also called anabolism. So here is the enzyme. This is the active site. That active site shape is perfectly complementary to the surface of these two substrates, A and B. So those substrates fit into that active site perfectly. So they become enzyme substrate complex, and then the reaction occurs. This reaction is about joining together so that these two substrates are joined together and become a new substance, AB. When the new substance AB is formed, it is released from the active site of the enzyme. Then the enzyme is free to take part in another reaction. So this is about joining together. So the second row is about breaking down. This is the enzyme. This is the active site. The shape of the active site is perfectly complementary to the shape of the substrate. So the substrate fit into the active site and they become enzyme substrate complex. This is about breaking down. So the substrate is broken down at this point. So two products are formed. After the reaction, these two products are released from the active site and the enzyme is free to take part in another reaction. So this is how enzymes work. So let's see about the investigation of the effect of temperature on amylase. Now, let's look at the investigation of the effect of temperature on amylase. To get started, we have to place the same amount of or volume of starch solution into 3 test tubes. Here, this is the example of the investigation. If you have to write your own design of investigation in your exam, you can use at least 2 or more test tubes to write this design of investigation. Here, I give you an example, 3 test tubes. And heat them up to 3 different temperature settings. For example, 30 degrees Celsius, 40 degrees Celsius, and 50 degrees Celsius. I use these examples because we have to keep in mind of the optimum temperature of the enzyme, which is usually around 37 or 40. So I use these 3 temperature settings to test one temperature lower than the optimum temperature and another temperature higher than the optimum temperature. And then you use the same number of drops of iodine and add them to the wells of the spotting tile that you can see here. It is a spotting tile. There are wells. After that, you add the same volume of amylase to all the test tubes that contain the starch solution and mix them well. After 10 seconds, you use a pipette to place 1 drop of mixture onto the first drop of iodine. Here is the first drop of iodine. So after another 10 seconds, you add another drop of mixture onto the second drop of iodine, which is the second well. So you keep doing that every 10 seconds until the iodine solution remains orange-brown, which is the original color, which means there is no more starch in your mixture solution. The starch is completely broken down due to the amylase. When that happens, you record the time taken for the reaction. And this whole investigation has to be repeated for reliability, to calculate average, and to remove anomalies. I write these reasons down here for your exam question, and this is really important for you to remember. Then we get the results, and we plot them down on this graph. So the results will give us a bell-shaped curve, just like this one. So here you can see at 15 degrees Celsius, there is no chemical reaction. And after that, as the temperature increases, the rate of reaction increases and reaches its highest point here at around 54 degrees Celsius. The reaction occurs fastest. After that, the rate of reaction slows down again and stops again at 75 degrees Celsius. When you plot the temperature against the time taken, you will get a reverse bell-shaped curve. I put this graph here because this is the usual tricky question that you will encounter in your exam paper. So you have to look at what is the value on the y-axis carefully. If it is rate of reaction, you will get bell-shaped curve. If it is time taken, you will get reverse bell-shaped curve, just like this one. So here at 5 degrees Celsius, the time taken is longest, which is 55 seconds, because if the reaction goes slowly, the time taken will be longer and longer. So this is the slowest reaction. When at around 38 degrees Celsius or 37, the reaction happens fastest. It took only like 19 seconds here. So this is the fastest chemical reaction. So you have to be able to read this graph really well. So this is the explanation why the results are like that. So enzymes are proteins, as I mentioned in the first slide, and as they have a specific shape. This is extremely important that the active site, the shape of the active site has to be specific shape, just like the shape of the substrate, so that they will fit into each other very well for the reaction to proceed. Enzymes work fastest at their optimum temperature because they have the highest kinetic energy and the most suitable temperature. For example, for our human body, it is 37 degrees Celsius. That is why our human body normal body temperature is 37 degrees Celsius. Heating the enzymes beyond the optimum temperature or any other proteins beyond the optimum temperature will cause that enzyme or protein to be denatured, which means that the substrates cannot fit into that denatured enzymes because the shape of the active site has been lost. So heating beyond the optimum temperature can cause the disruption of some chemical bonds in that protein so that the shape is no longer the specific shape that is complementary to the shape of the substrate. So here, I will show you the example. So here you can see this is the enzyme. So this is the shape of the active site, which is square, so that it is complementary to the shape of the substrate. But when we heat it beyond the optimum temperature, it could become rounded. So the square-shaped substrate can no longer fit into that rounded active site. So the reaction is not catalyzed anymore. This denaturation is irreversible. Once enzymes are denatured, they cannot regain their proper shape and their activities will stop. Now let's look at the investigation of the effect of pH on amylase. It is quite similar to the one before. So let's read this. We have to place the same number of drops of iodine solution into the rows on the towel, which is the same spot in towel. Use as arranged to place the same volume of amylase into 3 test tubes. This is the example. You can use 3 or 4 or 5 or even 2. And then add the same volume of 3 different buffer solutions. For example, pH 6, 7, and 8 to the test tubes. pH 7 is the usual optimum pH. That's why I put it in the middle. And then add the same volume of starch solution to the amylase test tubes and buffer solution, which means they are mixed. So mix them very well and start the stopwatch. After 10 seconds, you use a pipette to place 1 drop of that mixture onto the first drop of iodine here. You repeat this every 10 seconds until the iodine solution remains orange-brown, which means there is no starch in the mixture. The starch is completely broken down by the amylase. Then you record the time taken. This whole investigation has to be repeated just like the one before for reliability, to calculate average, and to remove anomalies. So the results are plotted, so you will get a bell-shaped curve. The optimum pH for most enzymes is 7, but some have a lower optimum pH, which is usually pH 2 in stomach, and some have a higher optimum pH, pH 8 or 9 in small intestine. So you can see those examples in this graph. So the middle one, the green curve, is for most enzymes. So the highest reaction rate is found around pH 6.5. So it can be rounded up to pH 7. So this is for most enzymes. The pepsin, which is the enzyme in the stomach, the highest rate is found around pH 2. The blue curve, this is for pancreatic lipase. The highest reaction rate is found around pH 8 and pH 9. So it is the optimum pH for pancreatic lipase, which is found in the small intestine. So the optimum pH could be varied. Moving too far away from the optimum pH will cause the enzyme to denature, and so the activity will stop. So just like the temperature. This will change the shape of the active site, so the substrate can no longer fit into it, reducing the rate of the reaction. So this is the end of this video. I hope this is helpful for you, and if you like this video, please like and subscribe our YouTube channel. Thank you for watching, and see you in next video. Bye-bye.